My name is Paige Wineinger, and this is the story of a mind-altering fungus called Cordycep unilateralis and its evolutionary relationship to the carpenter ant. So, what is an ophiocordycep? It is a strand of parasitic fungus living in the rainforest. More specifically, the strand that we will be talking about lives in the rainforests of Brazil and Thailand. The ophiocordycep is a parasitic fungus because it relies on a host species, like the carpenter ant, to reproduce. The cordycep infects the ants through mind alternation termed parasite manipulation. This means that once the host is infected, the parasite controls the mind and actions of the host. Now, each cordycep infects only one species. The Cordycep unilateralis infects the carpenter ant and no other organism. It infects the host by dropping spores to the forest floor. Spores are microscopic atoms that the ants walk over to become infected. This can be a difficult process for the cordycep because the carpenter ant colonies live about 80 feet into the canopies of the forest, while the cordyceps thrive only 1 foot from the forest floor. Additionally, the ants rarely ever descend to the floor because of the risk of infection. Only rarely do scavenger members of the colony descend when food is needed. When the ants do descend and become infected with the spores, parasite manipulation occurs. Remember that parasite manipulation is when the parasite controls the mind and actions of the host, in this case the carpenter ant. The spores penetrate through the exoskeleton of the ant and begin to deteriorate the unnecessary soft tissue. Then, through unknown mechanisms to scientists, the spores make their way to the brain and begin to take over control of the entire body, forcing the ants to climb branches and attach themselves to the main veins of the leaves. This causes the infected ants to be called zombie ants, because much like the zombies we see in the movies today, they are mindless, brainless creatures that appear to bumble around with no purpose and with no control. Now, once the ant has attached itself to the leaf, it dies, and the parasite begins to grow out of the top of the head. This growth is called a stroma, and it is what produces and drops the spores needed for the Cordycep unilateralis to reproduce. Here in this image, we see an infected host with a fully matured stroma growing from its head. The ant's grip on the vein is so strong that the marks can be found in fossils millions of years old. So, let's take a look at some studies and experiments conducted on this special relationship between the Cordycep unilateralis and the carpenter ant. The first question: how has the reproductive process and success of the Cordycep unilateralis co-evolved with the carpenter ant? Here, the scientists are looking for evidence of how the two organisms have evolved together over time. First, the scientists conducting this experiment hypothesized that in response to the ants' infrequent descent to the forest floor, the cordycep produces its spores slowly but releases them frequently during the lifetime of the stroma for an increased probability of infection. They also predict that this will also reduce the parasite's own risk for infection. This idea that the parasite Cordycep unilateralis can be infected by another parasite is known as hyperparasitism, and scientists have discovered that this is a rather high risk for the cordycep. So how are these scientists testing this hypothesis? Well, they collected 31 infected ants from a graveyard site in a Brazilian rainforest. Now, a graveyard site is an area close to the forest floor with high densities of dead, infected host ants from the Cordycep unilateralis. These ants were then placed on a platform just outside of the graveyard site, and the scientists recorded the spore discharge every morning for 3 days. The specimens were then taken back to the lab, where they were placed in a petri dish for 18 hours and stored at 100% humidity. And again, scientists continued to record the spore discharge every morning for the next 6 days. As this data was collected, the scientists observed each phase of the cordycep's reproductive process. It is broken down into 3 phases. Stage 1, when the ant is freshly infected and killed, lasting on average 7 days. Then stage 2, when the stroma begins to grow and mature, lasting anywhere between 7 and 30 days. And finally stage 3, when the spores are produced and released to the forest floor, lasting on average 4 weeks before the stroma dies. Scientists also recorded the parasite's survival rate within each stage, noting when hyperparasitism set in, if it did, when it did not, and how successful the reproductive cycle was for each stroma. If we take a look at this next slide, this is just a simplified diagram of the life and reproductive cycle of the Cordycep unilateralis. But in the bottom right-hand corner, you can see this diagram of where the carpenter ant colonies are relative to the fungal zone and why this can be problematic for the cordycep if it wants to successfully infect the host and reproduce. It's such a long distance away, about 79 feet. After the data was recorded and analyzed, the scientists found that their hypothesis was proven correct. The cordycep produces spores slowly but releases them frequently to increase the chance of infection. This is especially beneficial because scientists found that there is a 60% chance of infection due to hyperparasitism during the second stage, the stage of stroma growth. However, there is only a less than 6% chance of infection due to hyperparasitism during the reproductive stage, which means that when the parasite reaches the reproductive stage, it frequently drops a lot of healthy, active spores that have a chance of infecting a host. Additionally, the scientists confirmed that each spore only lives about 2 days before becoming infertile. This is also another adaptive trait for the Cordycep unilateralis to further prevent its own infection. So, the cordycep releases spores with short lifespans, but releases them frequently so there is a smaller chance of another parasite finding them and infecting them to cause infertility. The interaction between the Cordycep unilateralis, the carpenter ant, and the other parasites that can infect the cordycep is called the adaptive life history strategy. In a nutshell, the scientists have proven from this experiment that the cordycep has evolved to account not only for its own reproductive needs and the living patterns of the carpenter ants, but also the infection patterns of its parasitic predators. The Cordycep unilateralis has evolved with the carpenter ant as well as the other parasites that threaten its reproductive cycles. Let's look at another experiment. Scientists are looking to know if the environment has an effect on the infection and reproductive success of the Cordycep unilateralis. Like the last experiment, they begin with a hypothesis, the prediction that the reproductive success and frequency of the Cordycep unilateralis is related to biotic factors such as temperature and humidity levels. Basically, biotic factors are naturally occurring factors. With this prediction, the scientists hope to prove that higher temperature and humidity levels create an optimum environment for reproduction. So the test. The scientists went back to the rainforest and identified 5 different graveyard sites. Then, they counted the number of dead, infected ants in each site, also recording humidity and temperature levels as well as amounts of vegetation being how thick the leaves and trees are. This data was logged, and the scientists continued to visit the sites and record their data until they had collected enough to cover both the wet and dry seasons. Additionally, the scientists looked for any fossils that may indicate how these biotic factors influenced the reproductive success of cordyceps in the past. They successfully uncovered a group of 48 million-year-old carpenter ant fossils that had been infected with the same strand of cordyceps studied in this presentation. The specimens were taken back to the labs and the soil was tested for humidity and temperature levels. In this image, you can see the bite marks left by the ants in this fossilized leaf. They are identified by the green circles. Even just studying the soils and number of bite marks from this fossil will also give clues to the scientists as to how the humidity and temperature levels affected infection rates. So what do these clues and data reveal? That once again, the scientists were proven correct. Larger quantities of dead infected hosts were found in the graveyards during the wet seasons, times of higher temperatures and humidity, as well as thicker, lusher vegetation, showing that reproduction is much more successful during these conditions. Scientists also found the same results after testing the fossils. Absolutely all of the fossils uncovered showed signs of high temperatures and humidities. Not a single sample was found with disputing data, proving that wet season conditions are optimal for successful reproductive success. There is one extra bit of information the scientists discovered that was not anticipated when conducting the experiment. Now, each graveyard had different density readings of the number of dead infected carpenter ants, but during the wet season, the density readings leveled out. Graveyards with already higher densities did not fluctuate much, but graveyards with lower density readings virtually skyrocketed. What could this mean? Well, this could be an indication that evolution and the environment has basically put a limit on how much reproductive success the Cordycep unilateralis can actually have. This can be evolution's way of preventing the extinction of the carpenter ant. So in a quick conclusion, the Cordycep unilateralis relies on its abilities to adapt not only to the living patterns of its host, the carpenter ant, but also the reproductive patterns of other threatening parasites. It relies on high temperatures and humidities to frequently reproduce successfully. However, this process is controlled by the environment to prevent the extinction of the carpenter ant. Thanks for listening. Check out these sources to read up on your own for some more information about this evolutionary process and relationship.